everybody, and thank you for attending the first Congressional Biomedical Research Caucus event. This is our 23rd year presenting these to members and staff on the importance of supporting biomedical research and the biomedical research that goes on in their own backyards. So I thank you all for coming. I'd like to introduce one of the caucus's chair people, Congresswoman Jackie Spear. Jackie represents the California 12th District. And her support in her district is high, and it's only matched by her support for biomedical research. So we'd like to thank you for your support. A couple of just quick things I need to do. The Coalition for Life Sciences is the group that is putting this event on. We are 8 nonprofit professional organizations that run the gamut from biomedical and biochemistry to genetics to cell biology. We are working tirelessly here on Capitol Hill to support and to build support for biomedical research. The Biomedical Research Caucus is a bicameral, bipartisan caucus, which is chaired by, along with Congresswoman Spear, Congressman Bill Bright, also from California, Congressman Ben from Pennsylvania, and Congressman Holt from New Jersey. So with no further ado, Congresswoman Spear.
So good afternoon, and thank you all for joining us. How many of you have sleep issues? Well, today you're going to hear, hopefully, many solutions on how to deal with it. I want to thank all the organizations associated with the Biomedical Caucus, Research Caucus, who helped fund this effort. I'm proud to be one of the co-chairs. I'm very proud that we have so many rising stars from the University of California, San Francisco, who we are able to showcase here during the year, and today is no exception. Dr. Louis Patakic is the guest this afternoon, and he is going to be a very impressive person to hear from. He is a distinguished neuroscientist who has dedicated countless waking and probably sleeping hours to his work in the study of the body's natural biorhythms and the genes that determine whether we are early birds or night owls. And unfortunately, when we're both, we are in deep trouble. He has made significant contributions to the study of inherited disorders related to the nervous system. His goal is to understand aspects of normal brain function, including human sleep behavior and the molecular basis of diseases such as epilepsy and migraine headache. Ultimately, such understanding will lead to better treatments. He will also discuss his work to develop drugs that could possibly reset the human clock for individuals with certain sleep disorders or travelers to different time zones who experience jet lag. I have to leave, unfortunately, Doctor, to go to another event, but I'm going to ask you my question now, and hopefully in the course of your presentation, you will discuss it. There are a whole cast of 53 members from the California delegation that, for the most part, each week travel here to DC and back to our districts, and we are never on the right clock for our bodies. So I hope you will offer up some tips on how we can get through the week and still be sane and understandable. I also want to note that Doctor took the red eye here to be with us today, and just this weekend came home from Japan. So if anyone knows how to fix the clock in our bodies, it's him, and we welcome you this afternoon.
Thank you, Congresswoman Spear, and to the Biomedical Research Caucus. It's a great pleasure and an honor to be here, and I'm pleased to be able to tell you about some of the work we've done. So my group is interested in 2 big areas. One is episodic disorders, and we've cloned many genes for human muscular dystrophies, episodic movement disorders, not including Parkinson's disease, but that are relevant to Parkinson's disease. Epilepsy and migraine headache, and those are diseases, right? Disorders of the nervous system. And what's different about the work that I'm going to tell you about today is that these are normal human behaviors. And that's different than diseases where people are falling down and convulsing, having pounding headaches that make them lay down in a dark room, or having difficulties walking because of a movement disorder. And behavior is very difficult to study because we have to tease apart the things that we do, the part that we're trying to get at, the part that is biology and genetics, and we have to tease apart the 4 cups of coffee that I had this morning to try to be able to speak coherently today, or the glass of wine that you may have had with dinner last night, the familial, cultural, psychosocial things that impinge on what our clock is trying to tell us to do. And that is a very, very challenging and difficult thing to do. And yet, let me tell you that we, despite those challenges, have been successful. So we have evolved on this wonderful planet that turns on its axis every 24 hours. And during the process of evolution, and unless, well, no, I'm not going to go there. Never mind. We have evolved to survive and actually to do well, to prosper on a planet that has these approximately 24-hour changes in the light-dark cycle and concurrent changes in temperature as a result of the light-dark cycle. And as a result of evolution, all aspects of our biochemistry and our metabolism have evolved to survive and to function maximally. And so all things about our biochemistry and metabolism, I will tell you, are linked to sleep and to the circadian clock. Cell cycle regulation and consequently cancer are linked to the circadian clock. Sleep disorders, of course. Affective disorders and probably all of psychiatric disorders are linked to clock. Migraine, headache, asthma, stroke, myocardial infarction, heart attack, and everything else. Learning and memory, our performance, pain thresholds, et cetera, et cetera. And I'll come back to that. So the circadian system is really made up of a number of different things. First of all, there's entrainment, light coming into the body through our eyes. Also, temperature as well as feeding can reset the clock when we travel from Japan to San Francisco to Washington, et cetera. And then there's the clock ticking in our brain, but there are also, haven't quite got these buttons down yet. There are also clocks ticking in each cell in our body, and we can dissociate the clock in our liver by feeding in a restricted feeding paradigm in rats or mice, for example. But it's the clock in the brain that is regulating and orchestrating the clocks in all the cells in the body. And when we're functioning well and normally and happily in one time zone, it's the clock in the brain that's like a conductor of an orchestra that's getting all of the members of the orchestra who can keep time by themselves pretty well, but it's keeping them all together so that the orchestra is coherent and the music is beautiful. And then there's coupling of the clock to the pacemaker outputs, the immune system cycling, the cell cycle cycling, the sleep-wake cycle cycling, and all of the outputs, the physiological outputs that the clock is regulating in a circadian fashion. So there are conventional sleepers that, say, go to bed at 11:30 and wake up at 7 or 7:30. Biologically, I'm speaking. This is without an alarm clock, without coffee, without light therapy, and so forth. There are people who can't fall asleep until 2 or 3 in the morning, and then consequently want to sleep in, although they frequently set an alarm because they have to go to school or they have to go to their job. Then there are morning larks who spontaneously can get up very early and consequently want to go to sleep much earlier than the average person. And retinally blind people can actually, if we measure their melatonin rhythms or their body temperature rhythms, they can actually drift in time, even though socially they may be living on a 24-hour clock. And then if we lesion the suprachiasmatic nucleus, the structure in the hypothalamus deep in the brain, monkeys or rats or mice will actually become completely arrhythmic and sleep and wake at random times during the day. And then there is another situation that's known in humans, and that is called natural short sleepers. These are individuals who just biologically require less sleep than the average person to feel good and to function at a very high level. So, and then there are also natural long sleepers. Now you have to differentiate the natural long sleepers from lazy people. There are also lazy people. And it's interesting how we all tend to make, and I don't want to do this, but people tend to make judgments about people's sleep patterns. You're lazy if you sleep in, or we feel kind of macho if we stay up all night, or whatever. Or took the red eye and still got up, and I'm speaking to you now, and it's I haven't slept very much. But natural short sleepers and natural long sleepers are people who biologically require very little or very much sleep to function at a high level and to feel good. So what wasn't known, these in the general population, there's a normal distribution of people in all of these different aspects. The amount of sleep required, the morningness versus eveningness of people. These are all, and how easily you can travel across time zones. Some people get hammered, other people can do it with facility and feel pretty good. They're normal variations in our abilities to do this, and I believe as a geneticist that there are strong genetic components to this. But what no one had recognized was strong genetic traits traveling in families that would allow us to do what people had done in Alzheimer's disease, Parkinson's disease, to find those rare families with strong genetic variants that allowed cloning of a gene that allowed access to a pathway that could be studied in a laboratory that has really informed the biology of Alzheimer's disease in the general Alzheimer population, or Parkinson's disease in the general Parkinson's population. Until Chris Jones, a colleague of mine at the University of Utah when I was there many years ago, identified this woman in his clinic who was 69 years old at the time, and she said, I've been going to doctors, they never listen to me, they tell me I'm depressed. She was depressed because everybody kept telling her this. No one was listening to her until Chris Jones listened to her, and this is an important clinical lesson. And he said, I think we've got something interesting here, and it looks genetic to me, and you're a geneticist, maybe we should look at this family. We studied this family together, and lo and behold, it looked like a very strong genetic trait. And through careful study of these patients between Chris and I over a period of time, we came up with what we thought was a sensitive and specific diagnostic criteria for something that we called familial advanced sleep phase syndrome. So advanced sleep phase syndrome was known. That is to say, as people get older, there's a strong tendency to get up earlier and to fall asleep earlier on average in the general population. And this was called advanced sleep phase syndrome. The sleep phase was advanced relative to the solar day, is advanced relative to the solar day in an aging population. But this is not in old people, well, although she's 69, which is, today's 69 is yesterday's 40, I guess. But and none of you are that anywhere close to that yet. But this is in people who are young, and she has gotten even earlier and earlier as she's gotten older. So this is an autosomal dominant form of advanced sleep phase syndrome, which we called FASPS. We studied these people in the hospital with EEG and measured their melatonin and measured their quality of sleep, their sleep architecture, and the result of this was that their sleep quality and quantity were normal. Everything was advanced. Their melatonin their peaks, their temperature rhythm nadirs and peaks, their sleep times, their wake-up times, everything was advanced that we could measure. But when they were sleeping, it was a normal quality sleep, and when they were awake, they were well rested, and so their sleep quality and quantity seemed to be normal. And we performed in that older woman one of the modern-day cave experiments that you probably have heard about, and showed that her period. Her clock was ticking too fast, much faster than an average person, a full hour shorter than an age and gender match control. And we published this in 1999. So only 13 years ago, the only, at that time, familial form of circadian rhythm variant in humans was recognized. And I'll tell you how common this is in a little bit. As common as it is, it was only recognized to be familial 13 years ago. And the reason, in part, is that these people were being blown off by their doctors, say, oh, you're just depressed, stop bothering me, there's nothing wrong with you. And these people are not falling down with seizures, they're not dying of horrible neurodegenerative diseases, they don't have a severe medical condition, even though that woman was very troubled by her waking up, as she put it, when it's cold, dark, and lonely. And yet another person in the family with the same mutation in the same gene, one of her male relatives, feels virtuous for his being a morning lark because he's the early bird that gets the worm, and he's been a very successful businessman, and I think is pretty well to do because he's been so successful in the business world. And so this underscores that this really is a behavioral phenotype, and whether it's a positive or a negative thing or an indifferent or neither a good or a bad thing is really in the eyes of the beholder. And so all of the work I'm going to tell you now is work that's been done in collaboration with my colleague and professor of neurology, Ying-Wei Fu, also at the University of California at San Francisco, a really remarkable human geneticist. She's the woman who cloned Fragile X mental retardation, myotonic dystrophy, one of the Alzheimer genes, when she was at a company in Seattle, and now is at the University of California. And she has shared in all of the work that I'm going to tell you about henceforth. So the first gene we cloned in that large Utah pedigree, that 69-year-old woman that I told you about, is this 23 exon gene, and it turns out that it's a homolog of the Drosophila gene called period. And the mutation is a spelling error in the amino acid sequence of a serine to a glycine at amino acid 662 in this 1,000 base pair protein called period. And at the same time, Joe Takahashi, then at Northwestern University, had cloned a gene called casein kinase 1, an enzyme encoding gene in a hamster that has a similar phenotype to this human variant. And this was a spontaneous mutation in nature in the golden Syrian hamster. And interestingly, kinases phosphorylate, add phosphate groups to serines, and so we showed then that casein kinase 1 binds to the protein in the region encoded by these exons, and we then began to study phosphorylation of this region of the protein. And I'll tell you why this is important later, because this may have implications for treatment of jet lag and for shift workers. So this was the first gene in humans that, and a mutation in a gene in humans that causes a variant in the circadian system and causes this large Utah pedigree to be extremely, or certain individuals in the family to be strong morning larks. Now I can tell you, let me show you this part of the amino acid sequence of the protein. This is the serine that's mutated to a glycine in that family from Utah. And interestingly, there are 4 more serines every third position downstream from this glycine. And we know that CK1, casein kinase 1, likes to phosphorylate serine or threonine residues that are 3 amino acids downstream of serines or threonines that have phosphate groups on them. Phosphate moieties have negative charges. And so we speculated that maybe these serines were phosphorylated by casein kinase 1 after a negative phosphate group is added to this serine. And I should add at this point that in another family with FASPS, indistinguishable from the large Utah family, we now have a mutation at the second serine in this register of 5 serines. which further supports this idea. Terry Toe, the student who did this work, did some in vitro experiments, biochemical experiments in a test tube to look for phosphorylation. And you can see that in the wild-type protein marked in green here, every other lane, that when he ran the protein out on a gel after treating it with kinase that he made in a test tube, that he got a gel shift because this protein now has a phosphate group added to it by the kinase, and therefore it moves more slowly through the gel. And so it has this extra band that's farther up in the gel. The mutant protein doesn't. So this supports the idea that there are additional serines being phosphorylated downstream. And I'll get to why this is important later on. So this mutation in PER2, this protein encoded by this gene, causes defective phosphorylation. And we thought that maybe it was because of the negative charge. And so when we replace this serine residue with a different amino acid called an aspartate, which is an amino acid that itself is negatively charged, he was able to reconstitute this gel shift, showing that the negative charge is what is required to lead to downstream phosphorylation. So the details of this are not, the biochemistry is not critically important for you guys at this point. Just to show you that we're beginning to understand some molecular details about the biochemistry of this, of the post-translational modification of the protein. Now let me go back to the clinical aspects and epidemiologic aspects. Chris and I have further studied in a population-based way FASPS, and we now know that the prevalence of this trait in the general population, at least in populations of a Northern European ancestry. This is a population based in Utah, which is largely Northern European descent, is 0.3 to 0.5%. So it's not terribly common, but we thought that this was going to be exceedingly rare. And so FASPS, as defined by our very strict clinical criteria, occurs in 1 in 200 to 1 in 330 people. So this is not terribly rare, and yet it was only described by us 13 years ago. So it's been out there, and people just haven't seen it, in part because they haven't been looking for it, or because they've been telling these people that there's, the ones who are troubled by it and come to sleep clinics, that there's nothing wrong with them. So what have we learned from Drosophila? So why should we do research in fruit flies? We've learned a tremendous amount. We know that two proteins, clock and cycle, bind to these promoter elements and drive transcription of two genes, period and timeless. Period is the homolog of the gene that the human gene that I've just told you about, and that these proteins are translated. We know that these proteins are post-translationally modified by kinases like double time, which is a casein kinase 1 homolog in Drosophila, and casein kinase 2, and this targets them for degradation. So the proteins are made, but then they're degraded. Timeless is modified by Shaggy, a GSK3 homolog in Drosophila, and it's degraded. So it takes a long time for these proteins to build up in the cytoplasm. But around evening time, they build up to a critical level, and then they're able to move into the nucleus, where they interact directly with clock and cycle and repress their own transcription, turn off their own transcription. Let me go back here, sorry. Now we know that light acting through another protein called cryptochrome in a way that we don't understand yet can lead to premature degradation of that complex, and then that can entrain the clock by turning on transcription again. But it's this loop. It's the turning on of these genes, the making of the proteins, which then accumulate slowly because they're being degraded, their movement into the nucleus where they turn off their own transcription, and then the time it takes for them to degrade when the process starts all over again. That whole process takes about 24 hours. And so this, in a very simplified way, is the molecular basis of the 24-hour clock. Now, in mammals, it's much more complicated. First of all, timeless is of no known function in the mammalian clock. That actually is going to change. We have a paper that's under review at Science showing mutations in human FASPS, but it's through a different molecular mechanism. So timeless has been relegated to the twilight zone of circadian genes by circadian biologists in the mammalian clock. And there are 3 period genes instead of 1. There are 2 cryptochromes instead of 1. There are these new genes, DEC1 and 2. And so the mammalian clock is not completely conserved. Some of it is similar, some of it is different. But we know a fair amount about the mammalian clock from work from a lot of people, including Joe Takahashi, who's formerly at Northwestern, now at UT Southwestern. And the human story now is adding a lot to this story. Now, because we now know that FASPS is much more common than we thought it would be, we've collected many families and we've started sequencing candidate genes. And the second one that we published is one in which we found a mutation in casein kinase 1 delta, a homolog of the casein kinase 1 epsilon gene in the golden Syrian hamster that I mentioned. And I have to tell you, we now have another paper under review that reports a second allele, so a second mutation in a different FASPS family in casein kinase 1 delta. But this is an enzyme that phosphorylates the period protein and many other proteins. And it's in this small family from Vermont collected by Bob Shapiro. And this family, the affected people, look just like the people in the Utah family. They wake up very early, 4 o'clock in the morning, without an alarm clock from a young age. They want to go to sleep very early. If they let themselves, they'd go to sleep at 6 o'clock. But they struggle to stay up so they can spend time with their unaffected spouse and with their unaffected children and so forth. And so these people look just like the first family I told you about. And the mutation occurs at this highly conserved residue in the domain of this enzyme that is responsible for transferring the phosphate onto the period protein and presumably other proteins that are phosphorylated by this kinase. When we do biochemistry in a test tube, we show that this mutation affects the ability of this kinase to do its job, its biochemical job. And so these families look identical clinically, but they, at a molecular and biochemical level, it looks quite similar also. In the case of the first story that I told you, the enzyme is normal, but the substrate, the PER protein, is mutated, so without a phosphate on this residue 662, the kinase can't phosphorylate these downstream serine residues. In the second case, where the kinase is mutated in either the hamster, which is called the tau mutant hamster, or the human families with CK1 delta mutations, even if this first serine 662 is able to be phosphorylated, and I'll tell you about that later, now the kinase is mutated and it is unable to phosphorylate these 4 downstream residues. And so the end result is inability to phosphorylate these downstream residues, and the phenotype is FASPS. So we're learning a lot about the biochemistry, and the inability to phosphorylate this region is one way that you can get FASPS. So now we want to study in mice these same mutations so that we can do things that we can't do in humans. We can't get pieces of their brains. We can't lock them in a room and turn off the lights for 3 weeks and just give them unlimited food and then record their activity on a wheel, not legally anyway. I have to be careful. This is being taped. So we knocked out the CK1 delta gene, and the mice are born, but then they die immediately. So this is a really important gene. This is a gene that's been well studied in cancer pathways, and I told you that there are tight connections between clock and cancer and many other things. So what we do to make mouse models, and we also make fly models of the human mutations, is to take the gene and to engineer the human mutation. And we often put the human gene into mice. So we also have to put the normal human gene into mice to make sure that anything that we see is not the result of putting the human gene into the mouse. So we have the normal human gene in a mouse, then we have the mutant human gene in a mouse, and we compare them. And we measure their activity on a wheel, first of all, when the lights are on for 12 hours and off for 12 hours for a week or two, and then we turn the lights off altogether and give them unlimited water and food, and measure activity as a surrogate for wakefulness, inactivity as a surrogate for sleep. And let me just tell you, I don't have any data here to show you, but let me tell you that the casein kinase 1 delta mutant mice, wild-type mice carrying the wild-type human transgene, these are called transgenes when we put them, engineer them and put them into mice, is completely normal. It has a period of about 23.7 hours. We humans have a period that's a little bit longer than 24 hours, and we have to adjust every day because of light coming into our eyes. Mice have a period that's a little bit shorter than 23 hours, and they have to adjust every day because of through light coming in through their eyes. But mice have a period shorter than 24 hours. We have a period a little longer than 24 hours. The wild-type mice had a period 23.7 hours. The mutant mice had a period that was almost 23 hours. So short period we predicted would cause FASPS, just like in the one human subject that we did the modern-day cave experiment. Now I'm going to show you some really cool data from the period mutant mice. We made 3 different transgenic mouse lines. We made the human wild-type normal gene and put it into mice. We made a human gene carrying the glycine mutation that the human subjects have. But we also made one that has the aspartate mutation. That's the amino acid that has a negative charge and will look like a serine that is always phosphorylated. And we predicted that this one would actually have a longer period. Now, the wild type, the mice that have the human PER gene engineered into them have a period that's 23.7 hours. That's completely normal for these mice. So it doesn't change the period. That's the same as a regular mouse would have. This is a regular mouse plus the human wild type or normal gene. Here is a mouse that's carrying the human gene with the human mutation. And here's a mouse, and it has a period that's 22 hours, so almost 2 hours shorter than a normal mouse, control mouse. Here is a mouse that has a mutation that we've never found in humans, but that we predicted would have a long period. Now, many of my predictions turn out not to be true. I'm not always right, although I like to think that I am. But here's one time that I was right, and so this is so satisfying for me. This mouse has a period that's longer than 24 hours, the opposite of this one where you can never phosphorylate a glycine. This one with an aspartate there looks like it's always phosphorylated because the aspartate has a negative charge. And then when we cross these human mutant transgenes, the glycine and aspartate genes, onto a mouse where we've knocked out the normal mouse PER genes in the background, the period gets even shorter, 20.7 hours, and even longer, 25 hours. So this PER2 protein, I told you, has about 1,000 amino acids. And this one amino acid, when we can dial the charge on that amino acid from 0, because a glycine can't be phosphorylated, to -1, because an aspartate has a charge of -1, and then with the wild type being somewhere in between, because kinases and phosphatases are 2 enzymes that are fighting back and forth to phosphorylate it and dephosphorylate it. So in the case of a wild type mouse, the part of the protein is negatively charged at this residue and part of it is uncharged because it's unphosphorylated. One amino acid, whether it's charged or not, phosphorylated or not, can dial the period of these animals anywhere from less than 21 hours to 25 hours. I think that is absolutely remarkable. So this is a very important switch for dialing the clock in these animals. And I'll tell you in a moment why I think that, I'm building up to why I think that's so important and where I think we may have a chance to develop jet lag drugs. When we put these animals in a light dark environment where the lights are on for 12 hours, off for 12 hours, these are groups of animals, 8 and 8 and 10 and 10. The red are mutant animals, the blue are wild type animals. Remember, mice are nocturnal, so they're 12 hours shifted from us. They're sleeping during the light phase, running around at night. These are just like the human subjects. They're doing what no self-respecting mouse, normal mouse would do. They're running in the light when the eagles and the falcons will swoop down and eat them. They're running 4 to 6 hours before the lights go out, which is really not a good thing for mice to do in the real world. And then they run in the dark, and then they get off their wheels and presumably are sleeping here well before the lights go off. So these animals are doing just what the human subjects are doing. They're phase advanced relative to the solar light dark cycle, and they look exactly. Now they get on the wheels right at lights off when we put them on a day length of 21 hours. So it is indeed the period shortening that's leading these animals to have a phase advance, and we presume that that is also what's happening in the human subjects. So if these human subjects moved to a planet with a 21-hour day, the human subjects from Utah or the Vermont family, we presume that they would live very normally on a 21-hour day. We would have trouble with that, but not the families with these genetic variants. So this is, I'm going to show you some genetic experiments, but this is what we've found. Let me tell you the punchline. We've shown now through biochemical experiments that residue 662 is not phosphorylated by casein kinase 1, this kinase I've talked so much about. It's phosphorylated by a kinase that up until just recently we've proven exists, but we didn't know its identity. I'll call it the green kinase. But once this residue is phosphorylated, has a negative charge there, casein kinase 1 can swoop in and phosphorylate those 4 downstream residues, 1, 2, 3, 4, and that throws a switch that leads to increased transcription of the PER2 message and increased PER2 protein. All of the dogma in the field from Drosophila and from rodents before had taught us that phosphorylation of proteins leads to their degradation. And so what we've shown now is that low PER protein levels leads to short periods, high PER protein levels leads to long periods, and phosphorylation of this region leads to increased PER2 protein levels by increased transcription. This was antithetical. This was heresy in the field when we initially published this paper, and has now become well accepted. And this seems so wasteful to me, nature working against itself. But what I've come to believe is that nature has this one kinase working against itself and has this fantastic green kinase as a very fine control point to really dial very precise control of the circadian clock. And so this kinase is really a key regulator of circadian timing in the clock. And so there's a balance normally, and we have now identified this kinase. It's as yet unpublished, and this is a perfect target now for developing jet lag drugs or drugs, small chemical compounds that you might take if you were a nurse that worked the night shift, or if you're traveling across time zones, or if you were driving the Exxon Valdez and wanted to be wide awake so you didn't run it aground and was it Alaska and create this huge oil spill. A number of these and all of the motor vehicle accidents, many of them are alcohol related, but many of them are sleep related. So all of these problems that we humans face as a result of sleep deprivation or being out of phase with our clock. So, how many of you have watched The Matrix? This is my marketing scheme. You take the blue pill when you're flying to Europe. You take the red pill when you're flying to China. So if we have activators of this kinase, turn this kinase activity up, that's exactly what you would want if you're flying to Taipei or Tokyo or Beijing or Seoul or, if you're flying west. If you're flying to Amsterdam or Paris or Rome, you'd want to inhibit this kinase, and you would then be sort of like one of these people in the PER2 family, or the casein kinase 1 delta family. Temporarily, you'd advance the clock and you'd get more quickly onto Paris time, Amsterdam time. So we've just identified this kinase. Casey Hallow's a fantastic postdoc in the lab. And so we're going to be now screening a large chemical library. And anything that we find that inhibits or activates the kinase, we can immediately take into animals and see if we can, and into cell culture in the laboratory, and see if we can change the phase of animals or cells growing in a petri dish. And so we'll see. There are a lot of ifs here, but this is, we had been studying this for purely basic science kinds of reasons. One family in the entire world so far that has a mutation at this site that led us to focus on the biochemistry that I've spent so much time talking about today. But through understanding of the biochemistry of this region of the PER2 protein led to identification of this kinase, which now seems like a great target for developing jet lag drugs. I'm going to skip all of this in the interest of time because I do want to. These are genetic experiments that just support the model, and I'm happy to talk with any of you about this. But this is the idea. Therapies, we've gained a lot of knowledge that lead us to think that the green kinase is a great target. Now, I want to take you on a couple of little side stories that have been truly remarkable. This family, all 6 of these people have FASPS. All of them have a CK1 delta mutation. We have a second family with a mutation 2 amino acids away from this one. They also have FASPS. All of the mutation carriers in these 2 families also have asthma and migraine with aura. And we have a paper under review now showing that migraine with aura is caused by the 2 mutations in the CK1 delta gene. So that is very, very exciting and quite interesting. It's a completely separate part of my lab that's working on episodic disorders, but it's funny how these different parts of our lives and our work can sometimes intersect. My interest in episodic disorders is, quite separate from my research effort in the circadian stuff, but here they've come to intersect, and so we're very excited about this work. FASPS is very prevalent, much more prevalent than we thought. We now have 80 families. We've only found 10 or 12 mutations in the canonical candidate clock genes. And so the human subjects afford a fantastic opportunity to find genes that have, I have a huge respect and admiration for the work that's been done in Drosophila and rodents and Arabidopsis, all of the different model organisms. But human genetics has a lot to offer. Every organism that's being studied has limitations and also tremendous tools and things to bring to bear in the field. But most of the focus in model organisms has been on finding mutations that change the speed of the clock. But we have 2 papers now under review that show that you can get FASPS because of mutations that cause alterations in entrainment. And so I think we're going to be finding, we've now begun to do whole genome sequencing on 16 of our unexplained FASPS families. And I think we're going to find novel entrainment genes that the forward mutagenesis screens in Drosophila and mice haven't found. And there's also the issue of once you have a clock that's ticking at a normal speed, how is it coupled to the pacemaker output? So mutations in genes that are encoding proteins that are important for coupling to those physiological outputs. That's another area that's really a black box at this point. And I think that there's great opportunities for finding novel genes here in the human populations that we've been studying. Because we've been studying a different phenotype. These screens have focused on period mutants. We're looking at a different phenotype. We're looking at phase advance, which can happen as a result of shortened periods, as I've shown you, but as I've told you but not shown you, it can also happen as a result of altered entrainment machinery. And I predict, although we haven't shown that yet, could result as effective altered output coupling. And I just demonstrate that here. All of the focus so far has been on clock genes. And so these are untapped areas where human genetics is going to come to bear in a really fantastic way, I would predict. So now let me just shift very quickly to sleep schedule, sleep quantity and quality. Factors affecting sleep, we have to strip away all of these environmental and other things to get at the intrinsic biological genetic processes. I've mentioned this at the very beginning. And so shown in blue here is the clock ticking in our brain that tells us to sleep, wake, sleep, wake, sleep, wake. Or I guess sleep is up here. Sorry, sleep, wake, wake. But separate from that is something that we call the sleep homeostat. So sleep is separate from the clock. I want to make that point very strongly. Sleepiness is what happens. We don't understand anything about sleepiness, but we get sleepier and sleepier. We accumulate something that we call sleepiness, whatever in the heck that is, as a function of how long we've been awake. So even though here our clock is telling us to be awake, if we've been sleep deprived, we can still sleep because we've accumulated a lot of that stuff, whatever it is, sleepiness, even though our clock is telling us to be awake. And so that's what we call sleep homeostatic machinery or mechanism. And so we, as a function of being awake, we accumulate sleepiness. That stuff, whatever it is, dissipates as we sleep, and it accumulates as we're awake, and it dissipates as we sleep. And it's the combination of those two things, sleepiness and the circadian clock ticking in our brain. It's the sum of those two things that make us however awake or sleepy we are at any one time in the day. So those are two separate processes, but they overlap too. And it's remarkable. We spend a third of our lives on average sleeping, and we understand almost zilch about sleep. And it's so important for our immune system function, for our memory and learning, for our normal cell cycle regulation. Hopefully, we're not going to get cancer and all these other things that I've already mentioned. Here's a woman from Kindred 7340. She's wearing a wristwatch that instrument that measures movement. And so here, this is like a running wheel for a mouse, but this is a activity monitor for a human. So she's awake and moving much, much more and sleeping. And so we measured this over many days. So she's sleeping 6 hours a day. We're not measuring EEG here. We're just measuring rest time. But my colleague Ingwe, who I mentioned earlier, showed that a mutation in this gene in this small family traveled with the people in this family who sleep a lot less than the average person. And they've done this their entire life, and they feel well rested. This woman is 70-something years old. She goes out dancing 4 nights a week. She travels internationally, once a month because, and she does a lot of charity work. And so she's well rested. She's energetic. She's a go-getter. And we made mutations in this mutation, their mutation in a mouse, and did EEG in the mice, and the mice sleep less than the wild-type control mice. So this is the first gene in mammals that regulates sleep. What a remarkable thing. So this is a transcription factor. We don't understand yet anything about the molecular mechanism here, but this is now a finger hold, just barely a finger hold, that we hope to use to pull ourselves up to understand something about what are the genes that this transcriptional regulator is turning on and off. That would be one mechanism or one way that we might get into mechanistic insights about what's regulating human sleep. Because, gosh, I would love a drug that could, make me really wakeful and only have to sleep 2 hours a night. We now have a second family and have cloned a gene and a mutation. This is not published yet. For another family that only sleeps certain people in the family sleep only 6 or 6.5 hours, lifelong, feel well rested, very energetic, go-getters. But they also have severe depression and anxiety. And when we make the mutation in the mice sleep less, as measured by EEG. These people do very well with lorazepam, which is a benzodiazepine. When we do the best available assays in the mice for depression-like behavior, they have depression-like behavior. When we treat the mice with these kinds of drugs, they get better. So we think that this mutation may be causing not only short sleep, but also a depression-like phenotype. This obviously is going to require a lot more work. But again, you can see how we have the possibility of perhaps getting at other phenotypes that are not just sleep and clock related, but tie into some of these other phenotypes that I mentioned early on. So now I'm just going to sum up, but tell you we've got a lot of FASPS families. Many of them don't have mutations in the known genes, so we're off searching for new genes that will hopefully take us into new pathways, new areas of biology that will give us new insights into the human clock and human sleep. We now have 45 familial natural short sleep families. Only 2 of those have mutations. So there are lots of families in which we hope to find additional genes through whole genome, whole exome sequencing. And we've also recently started to collect familial delayed sleep phase syndrome families. These are extreme families who have strong genetic alleles for delayed sleep phase or night owl syndrome. So these people can't fall asleep until 4 o'clock in the morning, 5 o'clock in the morning. This is a real problem for folks because of jobs and school start times and other things. Much more of a problem for these folks on average than for the FASPS people. And so these will be an interesting group of families to study as well. So a real gold mine, I think. Opportunities to learn about the clock and about human sleep and the connections to the immune system and disorders of the immune system. We're interested in looking at phenotypes of learning and memory in all of our mouse models or engaging others who are interested in doing that. Some of our mouse lines have tumors, a lot more tumors than the average mouse lines we have in the lab. Again, there's just too much, for us to do. There are ties between sleep and, night eating syndrome, for example, with ties with metabolism, obesity, and diabetes, mood disorders, and other psychiatric syndromes. And I've already told you about one tie to migraine. And so there's just a wealth of stuff to do here. Not only just with looking for clock and sleep genes and trying to understand the biology, but then the connections to all of these other physiological outputs and connections of clock and sleep. So I hope I'm imparting to you some of the excitement that I feel about where all of this could go. And there's value in studying all of these different organisms. My own passion as a neurologist and in characterizing these newly recognized phenotypes has been to start with the humans, but as I've shown you, we've taken advantage of engineering, taking genetic variants in the human and engineering them into rodents. We also use the fruit fly in the laboratory. We also use cells in culture and biochemical assays in the lab. To try to understand complex pathways and really get a handle on the molecular bases of sleep and clock. I always tell students and postdocs that it's best to use whatever hammer and as many hammers to hit the nail as you possibly can. And each of these organisms and each of these tools that we can use has advantages for different kinds of questions that we try to ask. And then I'll show in my final slide some of the people who've done the work that I've talked about today. Ingwe I've mentioned, Chris Jones I've mentioned, Andy Charles has been a great collaborator on the migraine project, and then funding from NIH, Heart, Lung, and Blood, NINDS, and Howard Hughes Medical Institute. And I'll thank you for your attention and be happy to take questions if time permits.
Before questions start, I'd like to introduce Congressman Rush from New Jersey.
I thank Dr. Tacek for an interesting presentation. I'm Rush Holt, one of the co-chairs, and you've heard from Representative Spear earlier, and so I won't go on at length, except to remind those of you from congressional offices why the Biomedical Research Caucus exists. It is to remind you of what we get for our investment in the area. Fascinating, relevant, economically relevant, as well as relevant in public health and individual medicine. With that, let me point out that in the budget, as the President has presented for this year, overall funding for research and development, all kinds of research and development, of which NIH is one part, the funding will be less as a percentage of the federal budget any time in 5 decades. Wow, yeah, wow. And NIH, with the brief, very welcome ARRA spike for the last roughly half dozen years, has been essentially flat, with a little bit of up and down. So, as you consider the important work that these folks are doing, and I don't know how much of it is funded by NIH, but a significant amount, I suppose, it's worth remembering why we have this caucus meeting. So with that, I will turn it back to the professor for comments, and thank you all for coming. Thank you very much for coming.
Thank you.